Hi everybody, welcome back to my channel. This is Sagnick Dutta. I'm a second professional MBBS student at Bardwan Medical College and Hospital, West Bengal. From this video onwards, I'm starting out with a new series of videos where I'll be teaching and discussing about the zoology chapters included in your NCERT, which would be useful for you for your Neet 2024 examinations. Now, starting from Neet 2021, the biology subject in the Neet paper has been divided into two sections, the botany and zoology. But for few chapters, there has been no clear indications whether those are included in botany or zoology. For example, the cell biology, it is equally relevant for botany as well as zoology. There are a few chapters which are specifically included in zoology, for example, the human physiology. Moreover, human physiology is something which would be very necessary for you once you get into your medical colleges as well. So I decided to start with the human physiology section from this video onwards. And this video is particularly about the chapter body fluids and circulations. So, starting with the very first chapter, which basically deals with the cardiovascular system. Now, if you notice carefully, the chapter's name is not cardiovascular system, it's body fluid and circulation. Now, the question is, why is it being termed as body fluids and not the cardiovascular system? Because once you get into the chapter, you will find out that in most of the sections, it has been discussed about the blood and the heart and the cardiac cycle and the cardiac rhythms, etc. Now, then why is the name given as body fluids and circulation? Because apart from blood, there are a lot of other body fluids present in the human body. Now, if you can think of some other body fluids, they can be the lymph, which is also dealt about in about a paragraph in the NCERT as well. During our pre-medical preparations, we find out that most of the times we are being taught about blood as the body fluid. But once you get into medical college, you will find out that lymph and its associated organs like the lymph nodes and the spleen are equally clinically relevant as that of the blood and heart. Moreover, some biochemical tests are being performed on the basis of these lymph and its associated organ, which becomes very clinically significant. Thus, in this chapter, we'll be talking about blood as well as about lymph. Now, if you know and if you're much interested in studying biology and the human biology, you will find out that apart from blood and lymph, there are a lot of other body fluids as well. If you can think of the central nervous system, you will find out that there is another fluid present in the central nervous system called as the cerebrospinal fluid, that is the CSF. From the name itself, it is very clear. Cerebro means it relates to the cerebrum and spino means the spinal cord. That means the CSF is something which actually surrounds the brain as well as the spinal cord. Now, if you come to the respiratory system, you will find about the pleural fluid which is present between the two pleural layers. You can also read and study about the synovial fluid which is present surrounding the different joints in the body. Thus, in our body, in the human body, there are a lot of fluids. But in this particular chapter, we'll be discussing about the cardiovascular system specifically and its related fluid, that is blood, and we'll also talk and discuss about the lymph. Now, without much ado, let's get into my computer screen and start studying. Now, starting our discussion with the introduction of something called as the blood, which we talked about a little earlier. Now, what is blood? Blood is a special connective tissue. Now, what's the speciality about it? It is actually a fluid connective tissue. What do we mean by a fluid? That it flows, right? According to the knowledge of physics, fluid connective tissue is basically something, a connective tissue which actually flows. Now, going to its composition, what does blood actually is composed of? Blood actually is composed of a fluid matrix, which is known as the plasma, and there are some formed elements as well. Now, if we talk about the percentage composition of these two, the fluid matrix and the formed elements, the plasma constitutes about 55% of the blood and the formed elements constitute about the rest 45%. Now, talking about the composition of the plasma in particular, it consists of some water, which is about 90 to 92%. So now you actually know what gives the fluid nature of the blood. It's basically nothing but water. Apart from water, there are present some proteins which constitutes to about 6 to 8% of the plasma. Apart from these proteins, there are present certain minerals as well. Now, what are these minerals? If we name them, these are sodium, magnesium, calcium, bicarbonate ions, and chloride ions. These are the most specific composition of the plasma in terms of the minerals. Now, what are these formed elements? The formed elements are nothing but the red blood cells, the white blood cells, and the platelets. Now, talking about the proteins present in the plasma, what are these? The proteins present in plasma are in particular 3 in number, if we talk about according to the NCERT, and these are basically the major proteins that are present. First and foremost is fibrinogen, which basically helps in the clotting of the blood. Secondly, we have the globulins, which help in the defense mechanism of the blood. And third, we have the albumins, which help in maintaining the osmotic balance of the body fluids and blood in particular. Now, there's one interesting fact about fibrinogen, that because this is known as the clotting factor, if we remove this clotting factor from the blood, what happens and what we get is known as the serum. Now, this term serum is very important because once we move to a clinical aspect, this is something that we actually use for the biochemical tests. We collect the serum from a patient, that is, we collect the plasma without the clotting factors, and we test it for the presence of some abnormal constituents. Now, let us get into the details of the formed elements. Now, talking about the formed elements, as I spoke earlier, the formed elements are basically the red blood cells, the RBCs, that is, which are also known as the erythrocytes. There are white blood cells, which are known as leukocytes, and there are platelets, which are known as thrombocytes. Let us get into and start our discussion with the first and foremost formed element, that is erythrocytes. Now, if we talk about the number of erythrocytes present are about 5 to 5.5 million per millimeter cube of blood. Now, once we talk about erythrocytes, we all know that RBCs are nothing but red blood cells, which are red in color, and that is what it gets its name from. But the question is, how does it get its red color? The answer to that question is the presence of a substance called as hemoglobin. Hemoglobin is nothing but a protein which is very much rich in iron and is present in the RBCs, which gives it its color. Now, if we talk about the amount of hemoglobin present in the blood of a healthy individual, it is about 12 to 16 grams per 100 ml of blood. Okay, now these molecules called as hemoglobin, these are very much significant in the transport of oxygen in the blood, and that is what RBCs are basically made for. Now, once we know the reason and function of RBCs, which is the transport of oxygen, we get to know the reason behind its construction. What we mean by construction is, if you closely look at RBC, it's very much different from the other cells in the body. How is it different? It is different in its shape. Most of the cells in our body are either circular or oval or maybe rectangle, triangle or anything, but these are biconcave in shape. Secondly, most of the cells in the body have got nucleus, which is very much devoid in RBCs. RBCs are devoid of nucleus. And not only nucleus, it is devoid of any other membrane bound organelles. And the reason is pretty simple. You think wisely. You are making up an RBC to carry out oxygen. So you need the maximum space for the carrying of oxygen. Now, if you fill this minute cell with these cell organelles like nucleus, endoplasmic reticulum, Golgi bodies, and etc., then what will happen is we'll be killing up the space, and that is what it is not meant for. So we devoid it of the nucleus and any other membrane bound organelles, and thus causing up an increase in the space for its oxygen transport. Now, if we talk about the lifespan of RBCs, it's about 120 days. This place where these RBCs are destroyed is given the name of spleen. This is an anatomical structure present in the body of human beings, which is known as the graveyard of RBCs, where most of the RBCs are being destroyed after around 120 days. So our next topic of discussions is WBCs. In comparison to the RBCs, they do not have any hemoglobin. Their number varies to around 6,000 to 8,000 per millimeter cube of blood. So RBCs are more in number than WBCs. And leukocytes are short-lived than RBCs. Now, if we talk about WBCs, WBCs are specifically divided into two types. These are agranulocytes and granulocytes. Now, from the name itself, it is very clear that agranulocytes are those WBCs which do not have any granules, and granulocytes are those which have granules. Now, granules are the determining factors of differentiating WBCs into different groups. Now, if we talk about agranulocytes, these are specifically of two types, namely lymphocytes and monocytes. And granulocytes are divided into three types: eosinophils, basophils, and neutrophils. Right. If we talk about the percentage of these, neutrophils are the most numerous ones of about 60 to 65%. Basophils are the least to about 0 to 1%. Eosinophils are around 2 to 3%. And if we talk about lymphocytes and monocytes, the lymphocytes are about 20 to 25%, and the rest are monocytes. Right. Now we need to talk about the specific functions of these kinds of WBCs, starting with eosinophils. Now let us discuss about eosinophils. Eosinophils actually are bilobed nucleus having cells. If we see it under the microscope, we will find that there is a presence of headphone-shaped nucleus with two lobes. Now, what is the function of eosinophils? It actually helps in resisting infections, and these increase in numbers during allergic reactions. Fine. Now, getting back to basophils. Basophils are found in case of some inflammatory reactions, and they secrete molecules like histamine, serotonin, and heparin. Talking about neutrophils and monocytes, these have almost similar functions. That is, they are phagocytic in nature, and once there is any foreign body inside the body, these neutrophils and monocytes destroy the foreign organisms entering into the body. And talking about lymphocytes, these pertain to the immune system in the body. Lymphocytes are basically of two types, the T lymphocytes and the B lymphocytes. The T lymphocytes, the term T relates to thymus, and thus the T lymphocytes mature in the thymus. Whereas if we talk about B lymphocytes, these relate to bone marrow. Once you get into your class 12th, you would find out that B lymphocytes, bone marrow derived lymphocytes, are related to your humoral immunity, and T lymphocytes relate to the cell-mediated immunity. Talking about the last formed elements, that is thrombocytes, these are also known as platelets. They are derived from megakaryocytes. Megakaryocytes are some special cells present in the bone marrow which give rise to these cells called as the platelets. Talking about the number of platelets, it's around 1,50,000 to 3,50,000 platelets per millimeter cube of blood. Now, what are platelets necessary for? Platelets are basically necessary for the coagulation and clotting of blood. So, any disorder in the number of platelet synthesis would lead to clotting disorders in the body. Right. So with this, we come to the end of discussion about the formed elements. Now let us talk about the blood groups. Blood grouping is done on the basis of the presence of antigen and antibody. The antigens are present on the membrane of the RBCs and the antibodies are present in the plasma. There's a basic thumb rule. The antigens which are present on the membrane of the RBCs do not have their antibodies in the plasma of the same person. For example, if we have the antigen A, then we will not have the anti-A antibody in the plasma. On the basis of this, blood grouping is being done, and this system of blood grouping is known as the ABO system of blood grouping. According to the ABO system of the blood grouping, there are 4 types of blood groups: A, B, AB, and O. Now this A blood group has A antigen in its RBC, B has B antigen, AB has both A and B antigens, and O has no antigen on its RBC. Now this A blood group has the anti-B antibody in its plasma. The B blood group has anti-A antibody in its plasma. AB do not have any antibody in its plasma, while the O blood group has got both anti-A and anti-B antibody in its plasma. So from this discussion, it is very much clear that if we talk of the AB blood group, the AB blood group can receive blood from anyone and everyone from all other blood groups because it do not have any antibody present. Whereas if we talk about the O blood group, it cannot receive blood from any other blood group, but it can donate blood to any other blood group because it do not have any antigen on its RBC. The AB blood group is known as the universal recipient blood group, and the O blood group is known as the universal donor blood group. Apart from ABO blood group, there is another type of blood grouping which is done, which is known as the RH blood grouping. This is also based on the same concept, but here the antigen is named as the RH antigen. The term RH comes from the name rhesus, which is the rhesus monkey, from whose blood, for the first time ever, this type of blood grouping was done. So if the individuals have the RH antigen present, these are known as RH positive persons, and if not, then they are known as RH negative persons. Now, pertaining to this RH positivity and negativity, there is a special clinical case that we need to discuss, which is known as the Erythroblastosis Fetalis. Now, think about a scenario. There is a mother who is RH negative. She gives birth to a RH positive blood child. Now, this mother doesn't have the RH antigen. Now, if for some reason this RH positive blood of the fetus gets exposed to the mother, if for some reason, which is not usually the case because the placental barrier is not so much leaky that the fetal blood would get leaked to the mother's blood, but if for some reason this fetal blood gets leaked into the mother's blood, the mother would develop RH antibodies against the RH antigens. Because the mother doesn't have the RH antigen in its body, so it would find this RH antigen to be a foreign substance and would essentially make up an RH antibody. Now, once it makes up an RH antibody, what will happen in its next pregnancy is these RH antibodies can get into the RH positive blood of the fetus and destroy its blood, resulting in the death of the fetus and severe anemia and jaundice as well. This condition is known as the Erythroblastosis Fetalis. With this, you should keep in mind that the first child who was Rh positive is not much affected, but the second child who is Rh positive is very much affected, resulting in Erythroblastosis Fetalis. With this, we come to the next topic of discussion, that is the coagulation of blood. Now, if there is some kind of injury or trauma, what happens is there is a continuous flow of blood out of the body, and this cannot continue for long. For that, there is an inherent mechanism of the body which is stimulated and it stops the blood loss, and that is known as the coagulation of the blood. Now, when there is some injury or trauma, the tissues and the platelets are activated, which in turn release some chemical substances known as the tissue thromboplastins, which activates the enzyme known as the thrombokinase. Now, these thrombokinase in turn activates the prothrombin to get converted to thrombin. Now, this thrombin in turn acts as the stimulator for the next step of coagulation, where it activates fibrinogen to fibrin, which forms a meshwork around the affected site. Now, once this meshwork is created, some dead WBCs come and attach in there, resulting in the form of a clot or a coagulum, and thus the loss of blood is arrested. As a result, the loss of blood is stopped. Now, you can see very easily that there is an involvement, a lot of enzymes in this process. Now, once you get into much details of it, you would also find that there are a lot of different factors involved in this process of coagulation, which is not required at this level. But once you get into a medical college, you will find out that as a result, this process is known as the cascade process. Cascade means where every other enzyme, every other factor is involving and promoting every other factor, so that there is a continuous and faster process of coagulation. As a lot of enzymes and factors are related in the process of coagulation, this process is known as cascade process. And one more thing which you should keep in mind is this entire process is very much affected and supported by an ion called as calcium. Calcium plays a very important role in clotting mechanism, and it acts as an activator in each and every step of this process of coagulation. Now, I'd like to give you information. If you are really interested in knowing this process of coagulation, I have made a separate video, and that is for the physiology chapters of the first year MBBS student, but that is something very interesting. If you really like and would like to know about it, you can just go and refer to that video and see what this cascade process actually looks like. Now, with this, we come to the end of discussion about the fluid connective tissue known as blood. Now, we should actually get into more details of the circulatory system of the human body. Now, if we talk about the circulatory system, there are different kinds of circulatory pathways in the entire animal kingdom, if we see. Now, the major two portions and parts of the circulatory pathways are known as the open circulatory system, which is usually seen in arthropods and mollusks, because they do not have any blood vessels. Rather, they pass through large vessels and they open into some open sacs or spaces. Whereas in closed blood circulatory pathways, there is a presence of a network of blood vessels. As a result, they do not open into open spaces and sacs. Rather, they open into a closed network of blood vessels. Now, talking about the closed network of the circulatory system, this is present primarily in every other vertebrate organism. Now, if we talk about vertebrates, we include the fishes, the amphibians, the reptiles, and the birds and mammals. Right. Now, talking about fishes, they have a two-chambered heart. Amphibians have a three-chambered heart. Reptiles have a three-chambered heart, but there is an exception of crocodile, which has got a four-chambered heart, just like birds and mammals. Four-chambered hearts are those which have got two atria and two ventricles. Fact about two-chambered heart, that is those present in the fishes, is they pump out deoxygenated blood, which is oxygenated by the gills present in them, and they are supplied to the body parts, from where the deoxygenated blood is carried to the heart yet again, and thus it's a single circulation. In amphibians and reptiles, what happens is there is a left atrium which receives the oxygenated blood from the gills and lungs or skins that are present in them, and the right atrium receives the deoxygenated blood from the body parts. But they now get mixed into a single ventricle, which is then pumped to the body parts. If you see clearly, there is 2 cycles of blood pumping, but because they are getting mixed at a single place, this is said to be a partial double circulation or an incomplete double circulation. Now in mammals and birds, they have got a 4-chambered heart. That means they have 2 atria and 2 ventricles. Right. The respective atrium send the bloods to the respective ventricles. Now we'll be talking about this in the human circulatory system section in details, how this mechanism of double circulation works. But we have got this very much idea about the types of circulation and the circulatory pathways. There are 2 types of circulatory pathways, the open and the closed circulatory pathways, out of which the open circulatory pathways is present in the non-chordates like arthropods and mollusks, whereas the closed circulatory pathways are present in the vertebrates, especially fishes, amphibians, reptiles, birds and mammals, out of which fishes have 2-chambered heart, amphibians have got 3-chambered heart, crocodile with an exception have got a 4-chambered heart, rather the other reptiles have got a 3-chambered heart as well. Yes, we come to the end of a discussion of the part 1 of this video, where we have discussed the basics of the cardiovascular system, of the circulatory system. In the next video, we'll be dealing with the human circulatory system in particular. If you really like this video, hit the like button, subscribe to the channel and share these videos with your friends and whoever might need this video. Thank you.